speaker has 10 minutes for the presentation according to the schedule. Kindly asking you to respect this time, and at the end of the session there will be discussions of all our presentations. Let me introduce the first speaker, Burak Koçak, with the presentation Changes in Soil Carbon Mineralization under the Effects of Fungicide Cyprodynamics. Please.
Ekran paylaşımını verebilirseniz eğer sunumu açacağım. If you give the share screen ability, I will share my screen.
Hocam ekran paylaşımı share screen yazıyor.
Tamam, oraya tıklıyorum da host disabled participant screen sharing diye uyarı veriyor.
Tamam, hemen bakıyorum hocam. Hocam tamamdır.
Herkes görebiliyor mu?
Yeah.
Evet.
Good morning, everyone. I am Burak Koçak. I am a research assistant in Department of Biology in Çukurova University in Adana, Turkey. Today, I am going to present my study titled as Changes in Soil Carbon Mineralization under the Effects of Fungicide Cyprodynal. Cyprodynal was first applied on cereals in France in 1993. This fungicide has been used on cereals, vegetables, fruits, field crops, and ornamental plants as foliar application. Products containing cyprodynal were marketed in most European countries, North America, and Japan. Cyprodynal was also used in control of Hyperthyroides, Erysipeia subspecies, Pyrenophoria teres, Rhinosporium secalis, Septorionodorum, Botulis subspecies, Alternaria subspecies, Venturia subspecies, and Molinalia subspecies. Formulation is used in foliar applications, whereas water dispersible molecules or emulsifiable concentrates type. Cyprodynal was identified as an inhibitor of methionine biosynthesis in liquid cultures of Botulis cinerea. It was reported that pathway of cytithionin in filamentous fungi like Neurospora grassa and Aspergillus nidulans has been established as a major route of homocysteine and methionine biosynthesis. In this pathway, cyprodynal targets cytithionin beta-lyse that synthesize cytithionin from homocysteine. Secondary target of cyprodynal was the secretion of plant cell wall-degrading enzymes in pathogenic fungi. It is believed that these enzymes have an important role in the early periods of plant infection, and penetration of plant tissues by fungal mycelium was inhibited in the absence of secretion of these enzymes. Soil microbial activities are critical in decomposition of processes. However, intensive applications of chemicals used in agriculture negatively influence on these processes in many soils. Stability and productivity of soil ecosystems depend largely on soil microorganisms and their activity. For these reasons, determination of side effects of pesticides on soil microbial communities is very critical for soil health. Carbon mineralization is an important parameter that is adopted in international guidelines for ecological risk assessment and standard tests of pesticides and other chemicals. After pesticide applications, there were several studies that soil carbon mineralization was increased in field and laboratory experiments. Carbon mineralization is also an age-old reliable method for studying microbial activities in soil. Changes in carbon mineralization have also been used as a criteria for pesticide toxicity. The objective of this study was to evaluate the effects of cyprodynal on soil carbon mineralization. So we mixed the cyprodynal at recommended field dose and is 2 and 4 folds with the agricultural soil, and carbonic restorations were measured at 28 centigrade degrees for 42 days. It was hypothesized that fungicide cyprodynal would increase soil carbon mineralization. Soil samples that were not previously exposed to fungicide cyprodynal were collected from the top layer in research farm of Faculty of Agriculture in Çukurova University in Adana. Immediately after collection, soil samples were taken to the laboratory in dark plastic bags Homogenize air react at 25 centigrade degrees during 96 hours, and sieve to 2 millimeter to remove plant tissue and subject to physical. So what do we determine soil properties? We determine the soil texture, pH, calcium carbonate contents, organic carbon, and total nitrogen contents. And finally, we measure the field capacity of soils. For the incubation experiments, 100 gram soil sample were placed in a 750 milliliter incubation vessels and mixed with and without fungicide cyprodinol at the recommended field dose, and it's 2 and 4-fold. The amount used in the recommended field dose treatment was the recommended field rate for cyprodinol that was written in its etiquette, with the assumption that soil weight was 1.4 milligram per decade at the effective soil depth of 1 millimeter. 3 replicates were used for each treatment and control. Empty vessels were used as blanks. Final moisture of contents of both soils were adjusted to 80% of their own field capacity before incubation at 28 centigrade degrees for 42 days. We used sodium hydroxide for carbon dioxide trap. Cumulative carbon mineralization was calculated by summing up all carbon dioxide measured days until the end of incubation period, while its rate was calculated by dividing cumulative mineralized carbon by its soil organic carbon of control and all applications for 42 days. To gain a significant difference in analysis of variance was performed to determine the differences between treatments in soil carbon mineralization. So what are the results? As you can see in the table, we determined the soil as a loamy soil and slightly basic. Field capacity of this soil was determined 24.52%. Soil organic carbon was 1.81%. Total nitrogen of soil was 0.163%. And the rate of carbon and nitrogen was found 11.12. In general, all doses of cyprodinol increased carbon mineralization in the whole experiment. Significant differences between control and recommended field dose were found on the 6-day and 37-day, and these data were similar between control and RD2. I will show the graph after this slide. No significant differences were found between RD and RD times 2, except on 42 days. Carbon mineralization under RD times 4, those were significantly higher than control in all of major days, except on 12 and 42 days. As you can see in this graph, this is the graph of carbon mineralization between 1st and 12 days. All treatments were significantly higher than control in first 12 days. And in this second graph of soil carbon mineralization, this graph showed that carbon mineralization between 16 and 48 days. Generally, RD and RD times were not significantly higher than control, but there were found significant differences between highest doses of cyprodinol and control. The effects of fungicides on microbial processes and soil microorganisms depend on many factors. Some of major factors include physical, chemical, and biological properties of soil and nature, and the dose of fungicides applying at each time. Leo et al. In 2011 compared the half-lives of cyprodinol in soil under field and greenhouse conditions at the dosage of 1.8 gram active ingredient per hectare, and reported that pesticide cyprodinol degrade faster under greenhouse condition than field condition, either in soil. They found that the temperature and humidity were almost at fixable of 80% of according to field capacity, moisture, under the greenhouse condition. These results correlate within our research data. These experiments indicated that cyprodinol may be used as an energy source for soil microorganisms. Cumulative carbonizations, all doses of cyprodinol significantly increased soil carbonizations compared to control. In contrast, there were found no significant differences between RD and RD times 2. Increased percentage of soil carbonization were found 9.9% for RD, 10.5% for RD times 2, and 15.5% for RD times 4 compared to control. This is the cumulative carbon graph of cumulative carbonization. As you can see in the graph, all treatments were higher than control. Cumulative carbonization rate of carbonizations until 30th, 37th, and 42th days were given in figure 4 after this slide. This rate, in all these days, all cyprodinol doses significantly increased the rate of soil carbonization compared to control. This is a graph of rate of carbonization. As you can see, all treatments were significantly higher than control, especially the highest dose of cyprodinol was significantly higher than all treatments and control. The previous studies about cyprodinol were mainly formation, photodegradation in aqueous solution, and biosynthesis of reference standards by microorganisms. Although biodegradation of cyprodinol has been reported in Shokin et al. In 1997, almost no literature is available about the effects of cyprodinol on soil carbonization. This study is the first report about the effects of cyprodinol on soil carbonization. Results of the current studies indicated that the commanded field dose of its 2- and 4-folds of cyprodinol stimulated the ratio of carbonization in this agricultural soil, in this loamy soil. So what are the conclusions? It's concluded that the commanded field dose of fungicide cyprodinol significantly increased carbonization in a loamy soil. In addition, there were generally no significant differences between RD and RD times 2 doses. Significant increase of rate of carbonization in the loamy soil was highest at the dosage of RD times 4 in whole experiment. These results may give a suggestion of reasonable and safe use of the pesticide cyprodinol. However, for further confirmation, effects of high recommended dosage of cyprodinol on soil microorganisms should be investigated more comprehensively to eliminate its possible side effects in soil. Therefore, it is suggested that further research is required to trials with soils having different organic matter, texture, and pH, and with more fungicide type, dose, and incubation period under laboratory, greenhouse, and field conditions. Thank you for listening to me, and thank you for your attention. If you have any question, I will gladly answer.
Thank you a lot for your presentation and presentation of your scientific researches. Discussion will be the end of our session. So now let me introduce Arda Avjemi with presentation determination of the optimal culture medium for in vitro development of Festuca rubra as a spear rubra L.
Thank you, Daria. Hello and welcome again. Good morning to everyone who is participating and also listening to us. And I hope my voice is being heard nice by you. Here's my presentation. Today I'm going to talk about the determination of the optimum culture medium for in vitro development of Festuca rubra, subspecies rubra. By the way, I'm working as a research assistant, doctor research assistant at the biology department of Kocaeli University, Turkey. So let's start with a brief introduction of our study. This is red fescue, as you see in the figures. Red fescue, the other name, the scientific name of Festuca rubra, is a perennial dense turf producing.
Don't see your presentation.
Really? Okay.
Make demonstration.
Okay, sorry. Now I think I shared it. Can you see? Okay.
Yeah. And please, regarding time frame, 10 minutes.
Okay. Today I'm going to talk about the determination of the optimum culture medium conditions for in vitro development of Festica rubra subspecies rubra. I am starting with the brief introduction of my study. This is red fescue, as you see in the graphs, and it's a member of Poaceae family. So it's a perennial dense turf producing cool season grass, which is usually used in golf courses, home lawns, and for erosion control and beautification of recreational areas. This plant is an excellent soil binder and used extensively for stabilizing waterways, ponds, slopes, and banks. So it is used for controlling erosion. The species is also used as a turf for lawns, athletic fields, golf courses, as I said, and playgrounds. So this is a figure of the red fescue cultured in vitro. This grass is hardy, wear resistant, and shade tolerant. It's also drought resistant. But after once you establish it to the field, it requires ample moisturement. Red fescue is rarely seeded impure, but also used in a mixture component of a mixture of grasses. Instead, it's used with grasses and legumes for specific purposes, also such as lawns, erosion control, again, and recreational areas. And in shaded areas, red fescue is often used as a key grass because it's tolerant for shade, does not require so much sunlight. So the advantages of these advantages of the red fescue make it an attractive option among other grass species used for forage and lawn production. This is the inside view of our growth chamber, and these are the in vitro cultures of red fescue cultured on a different culture medium, which I will say later in the slides. So this is plant tissue culture technique I used. It's used for production process in vitro plant material and utilizing meristems and organs, which can be differentiated under controlled environmental conditions. The technique is also used alongside other plant biotechnology applications such as molecular breeding and disease-resistant plants. After genetic transformation studies, you need to use this plant tissue culture technique to generate the plant material from a single cell. So this is actually the last step of the transformation studies on plants. After a successful genetic manipulation, plant cells are cultured in vitro on a tissue culture medium to obtain the complete plant. So red fescue is one of the plant species that's subjected to genetic manipulation to improve its desirable traits. So therefore, it's important to determine the optimum culture medium to produce this plant successfully. The main component of a tissue culture medium is microelements and microelements, also vitamins and amino acids. So every plant tissue culture medium has a specific formulation consisting of these substances. The success of the in vitro propagation study is highly dependent on tissue culture medium composition. So different medium compositions should be tested to select the most favorable composition to propagate each plant species. Thus, this study aims to determine the plant's culture medium requirements for its efficient in vitro propagation, and also understanding the plant's optimum in vitro nutritional requirements would contribute to molecular improvement studies by efficiently enabling the researchers to develop complete plants from transgenic cell lines. Here you can see the mediums that I used in the study: Linsmeyer-Skoog's medium, Reiche-Skoog's medium, de Grief-Jacobs, and Schenk-Hildebrand medium. So this is the material and method section. Culture media is, you can see here, I used, and media supplements, sucrose and agar, 30 grams per liter and 7 grams per liter I used. I tested these parameters: germination rate, leaf and root elongation, and their production. And software used for statistics are SPSS and Excel. So this is the culture conditions listed below. This is my photo period, 16 hours per day, and 23 plus minus 1 centigrade degree temperature, and a constant temperature degree to culture my plants. And the photosynthetic photon flux density of illumination here, as you can see. So here are the results. To make you follow, easier follow the results, I put this graph here to make you see the ingredients of the culture medium clearly. So after I sterilized the seeds, by the way, I inoculated them onto this medium. So these are the results after I gained at the end of the culture medium after 30 days of in vitro culturing. So here is the germination rate. The percentages of germination rate did not vary, and they were statistically the same actually. Schenck and Hildebrand medium gave the highest germination percentage, whereas the seeds cultured on MS medium had the lowest germination rate. The culture medium components such as micro and micro elements and supplements such as plant growth regulators are decisive in seed germination studies, in seed germination success in such studies. So the current study is solely based on the testing of the effects of medium compositions, which means the differences in all developmental parameters are culture medium specific. So the highest germination rate obtained from Schenck-Hildebrand medium might be attributed to its rich potassium nitrate and phosphate and vitamin content, since the other media contain these components at lower levels. Okay, I will be quick as much as I can. So here is the leaf numbers. The highest number of leaves was found in Degraaff-Jacobs medium, whereas the Linz-Meyer-Scook medium gave the lowest results. And as you see, the Schenck-Hildebrand and Degraaff-Jacobs medium gave statistically similar results. So the drawback in supplying leaf growth of red fescue may be due to the lack of vitamins and vitamins such as pyridoxine and nicotinic acid in its formulation by LS medium. So this is why you can see the least leaf number in that medium. Here is the leaf length, a leaf elongation parameter, and the statistically similar result here I get from Schenck-Hildebrand and Degraaff-Jacobs medium. So the minimum leaf length was achieved in Linz-Meyer-Scook's medium, and the lack of the vitamins here, as you see, when you compare with MS and other media, you can see the lack of amino acid, glycine, pyridoxine, and nicotinic acid. It affects very much the leaf length of red fescue. Also, here is the root number. The maximum root number achieved in Degraaff-Jacobs and MS medium. Here is the interesting point is the MS medium triggered the root number, root production, and rhizogenesis. So there should be another mechanism behind this. So I can explain this. For example, say Schenck-Hildebrand medium is rich in vitamin content, and it gave lesser root production than MS medium. So this finding suggested the role of glycine in root production in red fescue. In general, plants supplied with amino acids generally have higher contents of sugars, proteins, and other nutritional elements. Here is the root length, root elongation parameter. Again, MS Murashik and Scook medium gave the best parameters result. So I can attribute this. Also, the amino acid richness of the medium. In biological systems, it has been shown that amino acids have a chelating agent of metal ions. So in fact, amino acids can act as ligands for metals with low solubility, such as iron and zinc, or they may be used either for nutrient mobilization from soil for their translocation within the plant. So here are the conclusions. At the end of the successful transformation studies, you need to use plant tissue culture medium and the technique for produce the complete plant. So when you're using these techniques on Festuca rubra plant, if you aim to foliar growth, so you have to use Daguerreophila and Schenkel-Derbant media. They should be preferred over the other medium to achieve a better foliar growth in the plant. And also, if you're focusing on the root growth, Murashik Skog and Daguerreophila media are more efficient in rhizogenesis in the same species. Here are the references for your interest, and thank you for listening me. We do not hear you, Daria.
Thank you a lot for presenting the results of your researches. And let me introduce Islam Siper with presentation genome-wide and gene-specific epigenetic analysis in Fusarium species. Please, you have 10 minutes according to the schedule.
Dear participants, welcome to my presentation. My name is Özlem Sefer. I am working as a research assistant at Yeniçeri University, and I am studying for a master's degree for Istanbul University. I will talk about our study genome-wide and gene-specific epigenetic analysis in Fusarium species. Firstly, Fusarium graminarium and culmorum cause head blight and crown root disease of small green cells worldwide. The epidemic of Fusarium disease has been increasing due to the changes in climatic conditions and crop rotation. Genetic analysis in important step is struggling with graminarium and culmorum. Detailed characterization of graminarium and culmorum has become important more than ever since these species are high levels of genetic and phenotypic diversity. These findings reveal that novel approaches in detailed characterization of Fusarium species are needed in the future. Graminarium and culmorum produce P-cotescens that are known and Fusarium seeds. There are missing points related to chemotyping and gene expression analysis related to P-cotescens synthesis. Novel approaches are needed in the obtaining reliable and precise data related to genetic characterization of Fusarium isolate. Also, subspecies or species complex member diagnostic investigation resulted in confusing data. Diversity at the epigenomic level has not been studied using current genetic tools. The investigation related the epigenetic and epigenomic characterization of Fusarium, culmorum, and graminarium missing from the literature. Methylation differences-based profiling analysis could be used to determine characterization of Fusarium isolate. The correlation between several important traits are important disease management, such as health, chemotyping, sample collection years, geographic region, could be constructed more efficiently as compared to data obtained from genetic analysis. In the studies aim to reveal the genome-wide and gene-specific differences in methylation profiles in Fusarium isolate from Turkey. It was aimed to open a new solution with data obtained by performing epigenetic analysis in graminarium and culmorum in cases where phenotyping, biochemical, and genetic analysis are not sufficient in combating disease. In this section, I will talk about which methods we use in our work and findings at the end of each analysis. Basically, 5 different methods are used: in vitro growth, linear growth rate, genomic DNA and clade analysis, gene-specific methylation, and gene expression analysis. Firstly, 22 graminarium and 37 culmorum isolates were provided from Samsun 19 Mayıs University Culture Collection, and graminarium H11 refers to the strain from Seoul National University Culture Collection. All isolates were cultured potato, dextrose growth, or agar at room temperature for 7 days. 25 square millimeter agar pieces, 7 days old culture were incubated 10 milliliter PGB and incubated at temperature. At the end of the incubation period, cultures were filtered and spores were counted by using formalin, and linear growth rates were measured by agar deletion technique. 59 fusarium isolates were subcultured in PDA, but 51 isolates were able to be grown effectively. You can see the table. Fusarium gramineum H11 reference stain was also used with positive sexual and asexual reproduction analysis. The lowest and highest spore production amount was detected in fusarium isolates. Here you can see the table obtained according to the measurements taken on the 4th and 7th days. Linear growth rate was processed according to the millimeter per day ratio. The average linear growth rate value, all isolates showing the efficient growth was determined. Genomic DNA isolation from 7 days old culture by DNA isolation kit. DNA quality was controlled with agar gel and quantity was measured photometrically. Gradient analysis was carried out non-digestive, HPI-1 digestive, and MSC1 digestive and HPI-2 digestive genomic DNA. DNA digestion reaction were performed according to manufacturer instructions. The reaction conditions and PCR components are shown in figure. PCR products were dissolved 2% agar gel electrophoresis at 100 volts for 2 hours. The dendrogram and genetic similarity analysis were carried by MVSP software. All isolates obtained with 10 of 17 primars rapid and credible analysis. However, 7 primers we are not able to give positive results. Average similarity was found 42% in rapid analysis of graminarium. In addition, HBr2 digested and MSP1 digested rapid reaction were resulted 48 and 49% average similarity in graminarium isolates. In Kumarium isolate, average similarity of rapid performed with non-digested HBI digested and MSP digested DNA were found. Gamma-based dendrogram based on the similarity matrix showed 2 major subbranches: non-digested, HBI digested, and MSP digested group profiles of both species. 10 to 2 and 47 were determined as close isolates in fusarium graminarium. You can see the figure, while 10F and 11F have high similarity in Kumarium. In addition, 3F and 7F is isolates of graminarium show the lowest similarity. Among the fusarium Kumarium isolates, 9 to 1 and F4 showed the lowest similarity. Methylation profiles of 7 genes that have function fundamental biological pathway, Kumarium and graminarium, were analyzed. Data of 7 genes were obtained from LCBI and primers were designed Primus 3 software. PCR was performed total volume of 25 microliters. You can see the reaction condition and content in the figure. PCR production compared according to the absence or presence of interesting bands. Expect length fragments of all 7 genes successfully amplified from non-digested DNA with gene-specific primers. However, PCR of digested DNA either with HPI or MSP results in different patterns with respective isolates and genes. 2 isolates, fusarium Kumarium 10F and 11F, show same amplification profiles with HbF digested PCR. You can see the table. Similarly, Fusarium graminarium isolate and 22 and 47 had same profile, HBI2 digested. All results of gene-specific methylation assay provided that except GS1, other 6 have type 1 and type 4 epigenetic profiling differences. Gene expression analysis, 2 graminarium and 2 columarium isolates show high similarity in PREDA analysis, and these were chosen for gene expression analysis. In addition, 2 isolates that have low similarity with these 4 isolates were included also. Total RNA molecules were isolated 7-day-old culture. Fungal samples was homogenized with 3 reagents. Total isolates RNA were controlled in agar-gel electrophoresis and spectrophotometry measurement. cDNA synthesis was performed from total RNA molecules. The qPCR reaction will be performed using cyber green fluorescent dye. And obtained CP values were used to fold change in gene expression calculation. Fold change in gene expression were obtained according to data CT normalization by Livak and Schmidtgen. Statistically, analysis was carried out with graphite 5 prism survey. As a result of the gene expression analysis, it was found that expression one-way ANOVA analysis provided that isolate of both species have significant differences MGB1 expression. Statistically, analysis of T5 gene expression showed that 11.5 isolates do not have any significance, but other 3 isolates must have significance. Real-time PCR analysis that gene revealed that Fusarium graminarium have significant differences. Consist with the epigenetic analysis result of 10F and 11F isolates have similar pattern in all genes. Expression profiling was also similar. Similarly, graminarium isolates 22 and 47 that showed high similarity in the epigenetic analysis, and genes except that expression profiling was also similar. As a result of all work, the evolution of genomic and epigenetic diversity in these 2 species was aimed in the study. The precise correlation was detected between LGR capacity, sample collection years, host characteristic was detected by PREDA analysis. Positive correlation between selected isolated 2 species were detected via gene expression and gene-specific methylation analysis for MGB1 and 3.5 genes. This study reveals that MGB1, 3.5, and URA genes may be useful detecting differences in epigenetic levels in species belonging to the genus Fusarium. The findings show that the PCR and qPCR based methylation analysis could be provide powerful tool for discrimination and comparison of fungal isolates. Generally, this study showed that the epigenetic or epigenomic differences could be adaptable to Fusarium isolates as well as other funding. Thank you for your attention. This study was supported to research.
Thank you a lot. Let me introduce our next speaker, Saikal Babushova, with presentation isolation of predatory fungi from environmental samples. Please, 10 minutes.
Good morning, dear participants. I am from Kyrgyzstan, and my presentation is isolation of predatory fungi. And I'm sorry for my bad English. I will try explain to share with you my research. Let me. Someone to delete my second
We can't see presentation.
Someone delete my second screen. Let me again to join because my device is not allow.
If you don't mind, maybe we can listen next speaker and then we will come back. To
Okay.
Sakal, are you with us? Maybe
Please,
we can listen next speaker. You will
Yes.
fix your problems and then we will come back to you, okay?
yes.
So please, let me introduce Sertan Çevik, the presentation effects of water lodging on some morphological, physiological, and photosynthetic parameters in body. Please, 10 minutes.
Dear President, first of all, I want to thank the organizing committee and everyone who contributed to this organization, because we know that it's not easy organize such an event. I want to present my presentation in Turkish, but I wrote it in English so everyone can easily follow that. And please note that I am willing to answer all questions after presentation. Thank you so much. Herkese günaydın tekrardan. Sertan Çevik ben. Mersin Üniversitesi'nde çalışıyorum öğretim görevlisi olarak. Yine arkadaşım Doktor Ayşin Güzeldeğer'le birlikte bu çalışmaları gerçekleştirdik. Uzun zamandan beri aslında biz çeşitli abiyotik streslerin bitkiler üzerine etkisini çok yönlü olarak çalışıyoruz. Son zamanlarda özellikle moleküler boyutu üzerine odaklandık ama çok fazla üzerinde durmadığımız bir konu vardı. Bu su baskınının etkisini çok merak ediyorduk açıkçası. Bu kongre için de böyle bir çalışma organize ettik ve bugün size bu sonuçları paylaşmak istiyorum. Arpa'da çalıştık. Arpa biliyorsunuz ekonomik olarak oldukça değerli bir bitki. Yani yaklaşık olarak yıllık 150 milyon ton civarında üretim kapasitesine sahip dünyada. Türkiye'de yine önemli arpa üreticisi ülkelerden bir tanesi. Yani 6,5 milyon ton civarında bir üretim kapasitesi var. Ama geçenlerde bir açıklama olmuştu. Stratejik bitkiler arasında artık yer alacağı söylendi, özellikle Türkiye'de. O yüzden üretim kapasitesinin arttırılması düşünülüyor arpanın. Biliyorsunuz arpa 3 ana sektörde yoğun olarak kullanılıyor. Bunlardan en önemlisi de işte hayvanların beslenmesinde. Yani dünyada %70, bizde %80'in üzerinde hayvanların beslenmesi için yine içecek endüstrisinde kullanılıyor. Son zamanlarda bu Türkiye'de çok yaygın değil aslında ama bu insan beslenmesi için de ek gıda olarak da arpadan artık yararlanabiliyoruz. Arpa tabii tüm dünyada dedik yani kültürü yapılan bir bitki. Dolayısıyla kültürü yapılan her bitki gibi işte sürekli ya da süreli olarak çevresel stres etmenlerine maruz kalıyor. Bu kadar stratejik bir bitki de söz konusu olduğu için de üzerine çok fazla çalışma yapılan bitki türü olarak ön plana çıkıyor. Bitkiler biliyorsunuz stres etmenlerine maruz kaldığında ciddi bir verim kaybına uğruyorlar. Bu %50'lere kadar çıkabiliyor. Yayınlanan raporlara baktığınızda ise yani 2050 yılına kadar bu insan üreme sayısının diyeyim ya da artış hızının %50 oranında düşmesi gerektiği, yine aynı şekilde tarımsal üretiminde en az %70 oranında artması gerektiği ortaya konulmuş birçok raporla ki beslenebilelim diye. O yüzden bu bitkilerin bu ürün kaybını azaltmaya yönelik ciddi anlamda çalışmalar yapılıyor. Tabii yapılan çalışmalar bitkilerin hayatta kalmasını sağlamaya yönelik değil, ürün kapasitesini nasıl arttırabiliriz diye. Bitkiler çünkü çevresel stres etmenlerine bu döngülerinin herhangi bir döneminde maruz kalabilir. Bunlar da onları öldürmekten tutun da ki bizim için önemli olan ürün kaybına neden olabiliyor. Biz de bu çalışmada bu abiyotik stres etmenlerinden su baskını üzerinde durduk. Arpa bitkisinde neler yapabileceğini morfolojik, fizyolojik ve bazı fotosentetik analizlerle ortaya koymaya çalıştık. Bunun için 5 gün boyunca arpa bitkilerini büyüttükten sonra 15 gün süreyle su stresine maruz bıraktık, su baskını stresine. Burada gördüğünüz gibi bu konteynerler içerisinde sürekli olarak kökleri su içerisinde kaldı arpa bitkilerinin. Bu 15 gün süreyle böyle devam etti. Ardından çeşitli morfolojik parametrelerini ölçtük ki bunlardan bir tanesi işte gövde çapıydı. İşte yaprak sayısı, bitkinin uzunluğu, işte yaş ve kuru ağırlıkları, bunlardan bazıları. Aynı şekilde yaprak su potansiyelini belirledik. Onu da basınç odasında oldukça hassas bir şekilde belirleyebiliyorsunuz. Onun dışında yaprakta işte klorofil içeriği aynı şekilde fotosistem 2 etkinliğinde belirledik. Böylelikle fotosentez etkinliğiyle ilgili de fikir sahibi olmak istemiştik. İstatistik analizlerini de Stronti testle yaptık. Şimdi bu 15 gün süre içerisinde bitkilerdeki hani morfolojik görüntü bu haldeydi. Aslında hani en başta da söylemiştim, biz uzun zamandır birçok abiyotik stresle ilgili çalışmalar yaptık ama morfolojik olarak bu kadar çarpıcı bir sonuç hiç elde etmemiştik. Yani kuraklık uyguladığımızda, tuzluk uyguladığımızda tamam hani gelişim bir şekilde baskılanıyor ama su baskınındaki kadar çarpıcı bir sonuç hiç almamıştık. Bu bizim için ilk oldu. Aslında literatürde bu ve buna benzer bilgiler de olmakla birlikte bunu arpa bitkisinde gösteriyor olmak bu kadar net bir şekilde bizim için de iyi oldu açıkçası. Sadece gelişime değil, işte gövde çapında, işte bitkinin uzunluğunda, yaprak sayısında, kuru ve yaş ağırlıkta ciddi azalmalar meydana geldi ki örneğin çapta %32, işte bitki yüksekliğinde %41 oranında, işte yaprak sayısında %25, yine yaş ve kuru ağırlıkta da yani %55 ve %39'la sırasıyla azalmalar meydana geldi ki bunlar oldukça çarpıcı değerler olarak ön plana çıkıyor. Literatürde aslında bununla ilgili çok fazla ama birbiriyle çelişen bilgiler var. Baktığımızda hani bu örneğin bitkinin yüksekliğinde, bitki boyunda neden bu kadar azalma meydana geldiğiyle ilgili genellikle işte bu kök civarında sentezlenen fitohormonların su baskını sırasında sentezlenmediği, sentezlense dahi oksijen eksikliği nedeniyle taşınamadığı ortaya konulmuş. Dolayısıyla bu fitohormon eksikliğinin özellikle büyüme ve gelişmeyi baskılayabileceği ile ilgili raporlar literatürde mevcut. Bu yaş ve kuru ağırlıkta da yine azalmalar meydana gelmiş. Bunda daha çok aslında bu element miktarındaki azalmalara bağlamış birçok araştırmacı. Yalnız yapılan çalışmalarda aslında topraktaki element miktarında bir azalma artış meydana geldi ama bunun taşınımı ile ilgili ciddi problemler yaşandı. Özellikle anoksik ortamda elementler toprakta bulunsa dahi bitkinin bunu kullanamadığı ortaya konulmuş. Çünkü zaten morfolojik görüntülerde de bu net bir şekilde çalışmamızda belirtilmişti. Bu bizi biraz şaşırtan bir sonuç oldu. Yaprak su potansiyeli, belki bunun üzerine çalışılmalı açıkçası. Çünkü yaprak su potansiyeline baktığımızda stresli gruplarda yaprak su potansiyelinin çok daha yüksek olduğunu belirledik. Sonra literatüre baktık. Hatta biz bu çalışmayı birkaç kez tekrar ettik. Literatüre baktığımızda da aslında kafa karışıklığımızı literatüre de yansıttığını gördük. Çünkü literatürde çeşitli bitkilerde işte bu su baskını stresine maruz kalan bitkilerde su potansiyelinin arttığı, azaldığı ya da değişmediği ile ilgili birçok rapor var. Biz detaylarına baktığımızda da bunun işte stresin süresine bağlı olabileceği ya da işte genotipe bağlı olabileceği ya da işte toprağın çeşidine bağlı olabileceği gibi çeşitli sonuçlar gördük. Ancak biliyorsunuz bu strese maruz kaldığında stomalarını da kapatıyor. Acaba sadece stomalarını kapatması mı buna engel oluyor? Bu tam olarak aslında bilinmiyor. Dolayısıyla bu konu aslında araştırmaya açık. Yani su baskınına maruz kalan bitkilerdeki yaprak su potansiyelindeki değişimlerin moleküler olarak incelenmesi önemli olabilir. Bu özellikle bu stres altında bu strese maruz kalan bitkilerde verimin korunması için önemli ipuçları buradan sağlanabilir. Çünkü literatürde de açıkçası bununla ilgili ciddi açıklıklar olduğunu görebiliyoruz. Tabii fotosentez düştü. Fotosenteze yönelik daha doğrusu parametrelerin düştüğünü görüyoruz. Özellikle fotosistem 2 etkinliğinin ve klorofil içeriğinin bu anlamda azaldığını görüyoruz. Biliyorsunuz zaten bu su baskını stresine maruz kaldığında bitki stomalarını kapatıyor. Stomalarını kapattığında da içsel karbondioksit miktarı azalıyor. Tabii içsel karbondioksit miktarının azalması fotosentezi doğrudan etkileyen bir etmen. Özellikle C3 bitkileri söz konusuysa. Ama sadece bu mu? Bizce sadece bu değil, çünkü klorofil içeriğinde de ciddi azalmalar meydana geliyor. Çünkü stomaları kapattığında içsel karbondioksit miktarının azalmasına yanında içsel oksijen miktarının da arttığını biliyoruz. Ki bu radikal oluşumu için bulunmaz bir fırsat. Oksijenin biliyorsunuz bu elektronegatiflik özelliği nedeniyle radikal oluşumuna oldukça açık bir molekül. Dolayısıyla ciddi anlamda radikal oluşumu özellikle bu yıkımlarda önemli olabileceğine biz inanıyoruz. Literatürde buna ait bir takım bilgiler var ama biz bu çalışma kapsamında buna bakmadığımız için net bir şey söyleyemiyoruz. Yani temel olarak tabii tüm parametreleri etkiledi. Morfolojik, fizyolojik tüm parametreler, yani muhtemelen moleküler parametreler de bunlarla birlikte etkilenmiştir. Ama biz özellikle bu yaprak su potansiyelindeki su baskınına maruz kalan bitkilerde yaprak su potansiyelindeki bu değişimleri açıkçası merak ediyoruz. İşin moleküler kısmının özellikle araştırılması gerektiğini düşünüyoruz. Hatta bununla ilgili de zaten çalışmalarımız var. Yakın zamanda başlatmayı planladığımız bir taslağı oluşturduk. Tüm dinleyenler için de aslında bu böyle bir soru işareti olduğunu da söylemekte fayda var diyorum. Dinlediğiniz için teşekkür ediyorum. Eğer sorularınız varsa cevaplamak isterim.
So thank you a lot for your presentation. Unfortunately, Sekal Bogushova can't join to us. So if you have any questions to each other, or our session is closed. Can you hear me? No?
I have no question.
I have also no question.
Yes, I have not any question.
So thank you a lot for participation. Thank you for presentation of your researches, and have a nice day. Thank you. Bye.
Thank you.
Thank you.
Thank you.
Bye.